Hello, my name's Aiden Willis. I'm going to be presenting ectopic volar fibroblasts induce histologic changes in non-volar skin. This is research I participated in as part of the Garza Laboratory in the Department of Dermatology at Johns Hopkins School of Medicine. I conducted this research under the guidance of my principal investigator, Dr. Luis Garza, and my lab manager, Dr. Sam Lee. Before I get into the results of our research, I'd like to give a little bit of background information on the research that we conducted. This research was conducted as part of the STUMP project within the Garza Laboratory, and the STUMP project seeks to alter skin characteristics at amputation sites to improve the quality of life for amputees. Typically, when an amputation is performed, non-volar skin, that's the skin that's found across most of the body, is grafted over the amputation site. However, this skin is not particularly durable. Therefore, when a prosthetic is applied, when the amputee uses a prosthetic, that non-volar skin experiences a great deal of pressure and shear that often results in chafing and degradation of skin at the amputation site. However, there are certain skin types that possess unique functional adaptations that make it more resistant to these physical stresses, such as volar skin. And volar skin is characterized by increased epidermal thickness due to a thicker stratum corneum and increased keratinocyte activity. The goal of the STUMP project is to alter the skin identity of this non-volar skin at amputation sites to effectively change the skin characteristics, make that skin thicker so it's more durable and less likely to chafe and degrade. Our lab seeks to do this by focusing primarily on fibroblasts. Fibroblasts are found in the epidermis. They produce collagen fibers, and notably, fibroblasts possess positional information from their original location. And there are fibroblasts in non-volar skin, but there are also, more importantly, fibroblasts in volar skin. And these volar fibroblasts help drive the increased epidermal thickness that allows volar skin to be more durable. Therefore, our lab seeks to transplant volar fibroblasts into non-volar skin locations to use the positional memory of these volar fibroblasts to alter the skin identity in these non-volar sites to produce thicker skin that is more resistant to pressure and shearing at these non-volar sites. And effectively, we're creating a callus in non-volar skin locations. To conduct this research, our lab recruited 40 subjects comprised of 19 women and 21 men between the ages of 19 and 63. Isolated fibroblasts from the sole and the scalp were amplified and injected into mismatched locations within the volunteers, as indicated by figure A. Typically, this location was on the inside of the thigh. And as controls, just vehicles and non-volar fibroblasts were also injected to these sites. After a 2 or 5 month post-injection period, the injection site tissue was excised for further examination through staining and protein extraction and amplification. The effect of injected volar fibroblasts on non-volar skin identity was quantified by measuring epidermal thickness, keratinocyte cytoplasmic size, collagen fibril length, and keratin 9 expression. The first result I would like to discuss is epidermal thickness. Epidermal thickness was measured through H&E staining and basic light microscopy, as shown right here with the images on the left. More importantly is the plot on the right, and we are most interested in the red and blue dots on the far right side of the plot. The red dots represent the data from injected sole fibroblasts. The blue dots represent data from injected scalp fibroblasts, and the black dots represent data from the controls. After staining and measurement, what we found was that epidermal thickness increased after injection of sole fibroblasts. We measured an average epidermal thickness of 106.6 micrometers compared to epidermal thickness of the control, which measured an average of 91.9 micrometers. We also observed that epidermal thickness the sole fibroblast injection sites was significantly greater than that at scalp injection sites, which measured at 96.5 micrometers. And what this shows is that there are characteristics within those sole fibroblasts that are unique and actually drive increased epidermal thickness greater than other non-volar fibroblasts, or even volar fibroblasts from other locations in the body. And this is in line with what we expected to find, since the sole is characterized by really thick skin that is very resistant to degradation when put under stress. The next piece of data I would like to discuss is the keratinocyte cytoplasmic size. What we observed was non-volar keratinocytes had an increased cytoplasmic size of 105.1 micrometers squared post-injection of the sole fibroblast compared to the controls, which had a cytoplasmic size of 94.6 micrometers squared. Similarly with epidermal thickness, injection of scalp fibroblasts also resulted in increased cytoplasmic size. However, this increase was still less than what we observed for injection of sole fibroblasts. And we were interested in keratinocyte cytoplasmic size, since increased cytoplasmic size may be indicative of the skin identity changes that we would expect to find by injecting these volar sole fibroblasts into the non-volar skin. As the volar fibroblasts from the sole retain their positional information, they would then act on the surrounding tissue, causing the surrounding cells to, become more active to produce the thicker epidermis and characteristics that are more similar to those that you would observe in, say, your sole or the palm of your hand. You normally find thick skin. Next, I'd like to discuss keratin 9 protein and mRNA expression. We were interested in the keratin 9 protein and mRNA because previous research has actually demonstrated that keratin 9 is actually controlled by dermal fibroblasts. Therefore, if we were to observe increased fibroblast activity, this should be correlated with increased keratin 9 expression in the injection sites. And again, in line with our previous results, we found that non-volar tissue injected with sole or scalp fibroblasts express significantly more keratin 9 protein and mRNA than non-volar tissue. What this is showing is that injection of these fibroblasts is in fact altering skin traits of the surrounding tissue. We're seeing expression of the protein that is controlled by fibroblasts, and we're seeing that expression of this protein is changing when we inject the fibroblasts from the different location, the volar location, into the non-volar site. Finally, I'd like to discuss collagen fiber length. Again, we have imaging of the actual tissue samples on the left and a plot on the right showing the actual results of measuring these collagen fiber lengths. And again, in line with our previous data, what we observed was that injection of sole fibroblasts into non-volar skin produced an average collagen fiber length of 8.7 micrometers, which is significantly longer than the average length of the control, which was only 7.5 micrometers. And again, this is in line with what we expected. We see increased keratin 9 expression, suggesting that the volar fibroblasts are altering skin traits. And since fibroblasts do produce collagen, we would expect these more active fibroblasts to produce longer collagen fibers. And again, this is what we see. We see increased collagen fiber length length when these volar sole fibroblasts are injected into non-volar skin. And it's these kinds of changes, these increased cytoplasmic size, increased fiber length, increased expression of the keratin 9 that we believe is actually driving the epidermal thickness or the increased epidermal thickness that we initially observed. So in conclusion, the purpose of the study was to demonstrate that injection of autologous volar fibroblasts in non-volar skins can drive changes in the characteristics of the non-volar skin, defined by increases in epidermal thickness, keratinocyte cytoplasmic size, collagen fibril length, and keratin 9 expression. What we observed was that injection of these volar fibroblasts into non-volar skin produced statistically significant changes in epidermal thickness, keratinocyte cytoplasmic size, collagen fibril length, and keratin 9. These results suggest that the volar fibroblasts do in fact retain their original positional information and can alter the characteristics of non-volar skin tissue when transplanted to a new location. Thank you for listening.